Good morning. It's May 29th, and this is your daily brief in science. Here's everything you need to know. A first-of-its-kind hepatitis B drug, Bepirovirsen, shows potential for a functional cure in international studies, with about one-fifth of treated patients maintaining undetectable virus levels for 6 months after stopping all therapy. Early data suggests lower baseline S protein levels may be linked to higher response rates, as researchers investigate why only some patients respond. Trials funded by GSK and Ionis compared weekly Bepirovirsen injections plus standard therapy against a placebo, and the control group did not achieve a functional cure. Regulatory submissions are underway with agencies in the US, Canada, Europe, Japan, and China, with decisions anticipated later this year that could open a new therapeutic pathway if approved. Chronic hepatitis B carries a high burden and, if Bepirovirsen proves durable and safe, could signal a major shift in disease management and outcomes. The potential public health impact is significant given the global burden of hepatitis B and the limitations of existing therapies. Current tools, safe vaccination, diagnostics, and antivirals remain essential, but access is uneven, highlighting global health inequities. Despite potential benefits, accessibility and affordability concerns loom in low-resource regions, along with possible side effects affecting platelets, kidney function, and injection sites. Experts view the results as promising but not universally applicable, noting reduced benefit for patients with cirrhosis or HIV, and questions about generalizability to all groups. Note: full article is behind a paywall and accessible to subscribers in this edition. The ultimate goal is to diagnose, link patients to care, and prevent progression to liver disease or cancer, with Bepirovirsen potentially becoming a cornerstone of future cure strategies. GSK holds the operational license for Bepirovirsen through a collaboration with Ionis, enabling development and potential commercialization. A new study proposes that homing pigeons have iron-loaded macrophages in the liver that act as magnetic immune cells, potentially linking liver signals to the brain to form a compass. The research finds pigeons rely on solar cues on sunny days for navigation, while overcast conditions scramble their magnetic sense, indicating multiple navigational cues at play. While the liver shows the strongest magnetic signal, other sites like the beak and spleen have been noted in related work, and experts caution that more research is needed to confirm the mechanism and its universality across species. Independent experts find the result plausible, but say magnetoreception may involve multiple senses or strategies depending on the task. The study provides DOI information to help readers access the original science article and related commentary. It collaborates across institutions including the University of Bonn, University Hospital Bonn, University of Duisburg-Essen, and the Max Planck Institute of Animal Behavior. Some researchers outside the study express skepticism about the causal strength of iron in macrophages interacting with Earth's weak magnetic field. Experts propose that one mechanism may dominate long-distance navigation, while another assists in destination finding, with context and precision shaping which is used. Cautious notes emphasize the need for evidence on superparamagnetism's role and warn that depleting macrophages could affect physiology or stress, potentially confounding results. Findings could extend to other animals lacking strong cryptochrome-based sensing or operating in low light, suggesting broader magnetoreception mechanisms. Caveats include concerns that liver-based sensing could be confounded by iron intake and that macrophages elsewhere might be affected by the experimental treatment. The story reflects debates in magnetoreception, acknowledging alternative theories such as magnetically sensitive molecules in the eyes and suggesting multiple strategies could coexist across species and contexts. A new study maps tumor TLS across cancers, showing they vary in maturation, location, and organization and shape local immune programs in tumor cell states. Researchers built a pan-cancer atlas by combining spatial transcriptomics with AI on pathology, revealing a maturation continuum from early TLS states to secondary follicle-like structures and distinct intratumoral niches. TLS maturation aligns with coordinated immune organization, including B and T cell zoning, dendritic networks, cytokine signaling, and interferon responses. The work highlights the value of integrating spatial multi-omics, AI, and digital pathology to enable real-time patient stratification and personalized oncology care. Authors advocate incorporating TLS features into future immuno-oncology trials as a stratifier or endpoint, and propose longitudinal sampling and refined TLS profiling to test causal mechanisms. A composite TLS score reflecting maturation and spatial context may serve as a biomarker to guide immunotherapy decisions and predict response. The study analyzes over 3,000 samples and classifies TLSs by maturation, cellular makeup, and spatial relation to tumor and stroma, forming a framework for TLS profiling. Researchers developed scalable AI pipelines to detect, profile, and classify TLSs from spatial omics data and standard pathology slides for rapid, scalable analysis. An AI framework can identify and categorize TLSs directly from routine HE slides, enabling scalable clinical TLS profiling across thousands of slides and external cohorts. A separate AI model trained on 3,071 whole slide images demonstrates the ability to predict TLS maturation directly from standard pathology. A new study shows researchers using truncated octahedron-shaped silver nanoparticles, or MECONs, tuned in shape and surface chemistry to self-assemble into nanoparticle superlattices that realize transitional structures along the Nishiyama-Wasserman pathway. Brown University and the University of Michigan stabilized a transient intermediate structural phase that had been predicted but not previously observed physically. They demonstrated stabilization of a fleeting intermediate phase between face-centered cubic and body-centered cubic arrangements using specially shaped silver nanoparticles. A comprehensive supplementary materials package accompanies the article with extensive figures, tables, and methodological details to reproduce or extend the experiments. Key collaborators include Uchen and Yasutaka Nagaoka of Brown, Tim Moore of Michigan, with theoretical and computational support from Glotzer's lab at Michigan. The research was published in Science and supported by multiple NSF grants and Department of Energy funding. The work reflects a multidisciplinary collaboration across chemistry, physics, and materials science under NSF and DOE funding. Computational modeling from Sharon Glotzer's group supported the experiments, showing ligand-coated MECONs naturally form configurations consistent with in-transition Nishiyama-Wasserman phases. Observations and simulations show that sticky ligands are essential for assembling into configurations matching the Nishiyama-Wasserman transition pathways between FCC and BCC. The team demonstrated that sticky polymer-coated particles stabilize the transient Nishiyama-Wasserman states at measurable conditions. The resulting superlattices exhibit high structural purity and stability, enabling direct study of transition pathways between high-symmetry lattices. The nanoparticle superlattices exhibit deep, strong light-matter coupling and room-temperature quantum optical phenomena, with electrons in silver vibrating in unison with light and showing potential for practical quantum devices. MIT researchers have created a catheter coated with carbon nanotube nanosensors that can detect the bladder cancer biomarker NMP-22 directly in the bladder, enabling earlier detection than traditional urine tests. The nanosensors use synthetic antibodies on carbon nanotubes that respond to NMP-22, which is often diluted or degraded in urine, allowing in situ detection. The device provides in situ 360-degree optical imaging with a miniaturized ball lens at the tip, mapping biomarker presence in 3 dimensions to localized tumors. The rotating tip illuminates and collects fluorescence to produce chemical images that detect and localize cancerous tissue. Future work aims to miniaturize components and integrate sensors into standard cystoscopes for outpatient office-based testing. Experts say this could herald a paradigm shift toward real-time, spatially resolved molecular imaging in oncology, and minimally invasive cancer surveillance. The approach highlights a broader move toward in-situ diagnostics that bring sensors to the disease site, potentially reducing monitoring costs and improving early treatment outcomes. Potential applications include integrating sensors with cystoscopes and adapting the platform to other cancers or diseases by swapping nanosensors for different targets. The study was published around May 27, 2026, with DOI 1.0638/S41565-02172-7. The MIT team is led by Michael Strano, with key contributions from Wonjun Yim and Ho Young Kang, supported by Koch Institute and NSF. Overall goal is more frequent, less invasive surveillance that improves early localization of recurrences, reduces unnecessary biopsies, and lowers healthcare costs. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.